You are listening to Armchair Healthcare, a podcast on pills, placebos, and policy.
Welcome to Armchair Healthcare. We're pleased to have as our guest Yuval Cohen of Corbus Pharmaceuticals, which is targeting the endocannabinoid system. Maybe we can start talking about what is the endocannabinoid system?
Hey, Max, thanks for having me on. So the endocannabinoid system is, in many ways, a fairly unremarkable biological system in the body. It's a system of G protein-coupled receptors. They're endogenous ligands known as endocannabinoids, and the enzymes that metabolize them. The biological function of it, though, is really interesting and very well preserved across animal life on the planet. What they do, simplistically, is they keep our brain healthy and they keep our immune system healthy. There are two GPCRs in this system, the cannabinoid type 1 receptor, and that one, Max, is found primarily in the central nervous system, and it has to do with neurological function. So, for example, when you engage with CB1, you have antiemetic activity, you have anti-epileptic activities. It changes your mood. It's a euphoric receptor. It will increase your appetite, etc. The second GPCR in the system is the cannabinoid type 2 receptor, and that one is found primarily in immune cells, pretty much every single type of immune cell, as well as fibroblasts. And what that GPCR does is, when you engage with it, is it tends to control or modulate inflammation and to halt and sometimes even reverse fibrosis. And the reason we have this system is this is a very, very ancient evolutionary system for recovering from trauma. So again, CNS engagement and immune system engagement.
Okay, great. And then beyond CB1, CB2, are there other targets that are interesting?
There's a bunch of what used to be known as an orphan cannabinoid receptors. They're known as orphan because no one quite knows what binds to them. There are also a bunch of other ligands floating around. The problem there is nobody knows what they bind to. But overwhelming, if you look at the academic literature, it's around CB1 and CB2, and then the type of ligands that bind to them. And if I can, there really are three types of ways you can engage these receptors. As I said, there are two endogenous ligands, two endocannabinoids, anandamide and 2-AGA. They are naturally occurring. We make them, again, when we are stressed, but they tend to be very short-lived and they're not very potent. Recently, Max, I think you saw, there was a really interesting story about a woman, I think in Scotland, who was, and this was a little bit exaggerated, impervious to pain and constantly happy. Which she had, which is a great quality of life. It turns out what she had a mutation that prevented her endogenous cannabinoids from being metabolized at the normal rate. So they would hang around for longer. But otherwise, the endogenous cannabinoids aren't particularly helpful for pharmaceutical purposes. The second source of ligands that will bind to these GPCRs is obviously the cannabis plant. These are known as the phytocannabinoids or the botanical cannabinoids. By far the most famous ones are THC and CBD. I live in Brooklyn, New York, and pretty much every second store is selling CBD. And this is obvious. These are biologically active compounds. There's no question about it. I think the debate is around what to do with them. What exactly is their biology and what is their potency? But we saw, for example, GW Pharma with Epidiolex, a very, very successful launch to a CBD drug for epilepsy. The third bucket, as it were, of ligands that interact with GPCRs like CB1 or CB2 are rationally designed novel chemical entities. This is what Corbus does. And that's where you can really apply sort of traditional pharmaceutical techniques of medicinal chemistry, biological knowledge, and just plain old chemistry to taking compounds. Some of them look like cannabinoids. Some of them look very, very different to cannabinoids. and customize the way they interact with these receptors.
So do you think the synthetic molecules that are kind of rationally designed, those have greater potential than the phytocannabinoids?
It depends for what. The phytocannabinoids tend to be active in the CNS. There is a drug called Marinol. AbbVie have been making it for, I'm guessing, at least 20 years. It's basically THC extracted from plants. And
it's from like 1985.
At least, yeah. That sounds about right. It's really an old drug. And Marinol is given to patients who are undergoing very, very powerful chemotherapy. And what it does is two things. It prevents them from vomiting. It's an antiemetic. And it gives them the munchies, which is important. You want those patients to have appetite. So phytocannabinoids can be very effective on CNS, where they tend to have far less efficacy, especially at any dosage that would make any sense, is on the immune system. And that's where there's a big gap. And that's where we come in. A couple of others doubtless will be coming in. For example, Janssen have a program on this pathway looking at NASH, which we're beginning to explore ourselves.
But you wouldn't want something to stimulate appetite to go after NASH, right?
Absolutely not. So the potential of going after this with rationally designed compounds is you can start to separate effects on CB1 versus CB2. You can start to separate effects on CNS versus the periphery. You can start tinkering, for example, with systemic formulations versus topical formulations. None of that can you do with botanicals. They're very, very difficult they are what they are. Mm-hmm. Let's start with that. They tend to like your CNS a lot, and they tend to be difficult to formulate.
But on the synthetic side, like people have tried to go after CB1, CB2 before, and I think probably the highest profile drug there was Acomplia by Sanofi for obesity, mm-hmm. where patients would lose some weight. I think it was a moderate amount of weight, but they had suicidality. And I remember at the time just thinking, okay, there's like 100 million people who smoke pot. And so why would you want to do the opposite? Because Acomplia was an antagonist of CB1.
Correct. That was an unbelievably traumatic event for the pharmaceutical industry. I'll go as far as to say that a company like Corbus really wouldn't be in business had Acomplia not happened, because that trauma about 2008, 2009 led every single big pharma to stop working on cannabinoids and allowed companies of our size to sort of come in and really start building territory in that field. And you're 100% right. First of all, CB1 is tricky. Agonizing it in the brain is a good thing in terms of euphoria, antiemetic, anti-epileptic. But it's an interesting GPCR because agonizing it in the liver is actually a bad thing. It causes inflammation. CB2 is much easier. CB2, you always want to agonize, and by agonizing CB2, you're always going to have an effect, which is a reduction of inflammation and fibrosis. So, again, not to overcomplicate this, but going after inflammation, we think, is a much cleaner way of going about it, and certainly presents a really, really big field for us.
Great. And then maybe you can just discuss kind of what are you focusing on right now?
So we have a number of programs. The most advanced one is a drug called linabacim. Linabacim is an agonist, a peripheral agonist of CB2. So it likes to stay outside the brain, and it likes to bind and activate CB2 in immune cells. And what we're doing with that one is we're going after inflammation and fibrosis in rare diseases. 3 of those are autoimmune diseases, and then one of them is actually a genetic rare inflammatory disease. The 2 autoimmune diseases that are most advanced, because they're both in a phase 3 study, are a disease called scleroderma, or systemic sclerosis, and then another one called dermatomyositis. Scleroderma is the most lethal of the autoimmune diseases, and that phase 3 reads out next summer. And if that data is positive, we will seek registration, and that will be the first drug ever approved for scleroderma, and that was about 90,000 patients. The non-autoimmune rare disease we're targeting, sort of the odd man out, is cystic fibrosis. So, at first you go, why do we need another cystic fibrosis drug? There are quite a few out there, which is 100% true. There are multiple drugs out there. But what we don't have for cystic fibrosis is we don't have drugs that target inflammation. We have drugs that target the mucus, that target bacteria, etc. And the ability to target inflammation and therefore reduce the amounts and frequencies of pulmonary exacerbations is actually very attractive. And we're incredibly grateful because we've been supported to the tune of $30 million by the Cystic Fibrosis Foundation. So that's sort of linabacin. That's our most advanced product, a drug candidate. We have another one, Max, which is totally different. It's a novel chemical entity, just like linabacin, very different structure. It binds to a cannabinoid receptor, except this time it binds to CB1, not to CB2. It doesn't bind to the immune system at all. It binds to primarily to liver cells, and we're not going after rare diseases at all. We're going after the opposite, sort of probably the least rare disease out there in America at the moment, which is fatty liver disease, NAFLD or NASH. That one's in the preclinic. It's going to be in phase 1 at the end of the year. And then we have an NIH program that will kick in to do a phase 2 study, an intramural program with NIH next year sometime. And if that data is positive, that will be really interesting. In that one, we actually have a big pharma competitor. As I mentioned, it's Janssen. They have a drug that in many ways is very similar. It targets CB1 in the liver for NASH, but it has a really interesting twist to it. Just like our drug, Max, we both try and avoid the brain so as not to have the issues that Acomplia had or rimonabant. We do so by having a small molecule that's sort of, beefed up and charged with ionic charges that should make it very difficult to penetrate the brain. They achieve the same result by having a monoclonal antibody, which typically don't like to go across the blood-brain barrier. So,
but you would need to either give like an IV or sub-Q.
Correct. That one is a once-a-month sub-Q. It's in phase 2, and it will be really interesting to see what they get out of it. The science is pretty robust, so we'll see which one of us actually succeeds. But it's pretty exciting.
Yeah, so you're both doing the inverse agonists of CB1
without going into the brain. So think of it as the rimonabant or the Acomplia minus the brain bit.
So would it work by I guess people eating a little bit less, because that seems to be like one of the better cures for NASH or better treatments.
I would argue that's a better cure for a lot of things in life. But
yes, diabetes, cardiovascular.
True, but the main cause, and so this is what was so interesting about rimonabant. Initially they thought about rimonabant, and you painted it very, very accurately. The idea was it would be the anti-munchies drug. It would be the drug that would kill your appetite. And it was potent. But what was interesting, it's not only that they lost weight. Their blood works got better. Their liver looked much better. There was even a small study that they did on NASH patients, I think only at about a dozen patients, where a whole bunch of outcomes, all the way down to liver enzymes, look much better without a necessary correlation to the amount of weight they lost. And so the conclusion that both we and Johnson & Johnson reached was, and together with a lot of academic research since then, was the main side of action, the main side of benefit for Acomplia wasn't actually the reduction of appetite in the brain. It was actually direct action on the liver. So if you take the brain out, and that was done in a whole bunch of animal models and knockout animals, etc., you got the same results, but obviously without any of the psychotropic side effects. So it sort of shifted radically from, hey, let's make them not be hungry, to, hey, let's focus on getting the liver to function better.
No, that's very interesting, because I don't think I heard that. I mean, I remember they had all those kind of ancillary effects, but that's typically what you see in anti-obesity drugs. Like you see that with, even phentermine, which is just speed. On the so your scleroderma program, you're going after diffuse cutaneous systemic sclerosis.
Correct.
So that's a pretty tough indication. Like, what's the mechanism there?
The mechanism of the disease or the mechanism of the drug?
Of the drug.
Got it. Because the disease, of course, is of unknown etiology. It's an autoimmune disease. We know that. The mechanism of the drug is we've seen a bunch of things. So it starts with obviously preclinical data as always. We saw very, very robust data in what's the bleomycin models, both the pulmonary bleomycin model and the skin bleomycin models. Those are classic models of inflammation and fibrosis. We also had very, very nice model of a mouse that was constitutively expressing TGF-beta. That's a fibrotic model actually without inflammation. And again, we saw very, very promising data. And then we moved to people, to patients. And in a double-blind placebo-controlled study, albeit a short one, it was 4 months and I think 44 patients, what we saw was the following. First of all, the drug was very easy to tolerate. So that's an important thing for this disease. But we saw changes, sometimes statistically significant, sometimes directionally important, in the primary endpoint, which is a composite known as CRIS, in its subcomponents, which have to do with skin and forced vital capacity and patient assessment and physician assessment and disability. A bunch of PROs, such as PROMIS 29, so that's important. And one of the things that was very satisfactory for me to see as a molecular biologist was biomarker changes in skin biopsies that were taken, Max. Both looking at histology, so for example, looking at extracellular matrix deposition between placebo and drug arm, or leukocytes between placebo and drug arm, and transcriptome analysis, which is always fun to try and analyze, a gazillion genes, but we actually saw about 2,000 genes moving up or down in a way that made a lot of sense. The mechanism is, again, CB2 is expressed on immune cells and on fibroblasts. So those are two biggies in terms of their effect in this disease. It's really what fuels this disease. And engaging with CB2 reverses that inflammation and tends to stop or reduce fibrosis. It depends on the tissue. So we had a pretty coherent set of data. That study is still actually ongoing as an open label extension study. Patients, I think we'll be showing the 2-year open label extension coming up at EULAR. Of course, remember, we no longer have placebo. Now it's just primarily safety and looking at the population within itself. And a phase 3 study. And that one, we just finished enrollment, I think 2 weeks ago. And data will be coming out next summer. It's a very large study for this disease. 365 patients, 1-year-long study, double-blind placebo-controlled with 2 doses versus placebo. And we think that if the data is strong, if we do have, again, robust data, then we've got a really good chance at registration.
And is it like generally the same design as the initial study in terms of like the doses you're using, et cetera?
Yeah, the doses are the same. Initial study had a sort of an escalation built into it. This one doesn't need to. It has 3 parallel arms, placebo, and then sort of a low dose and a high dose. We expect the low dose to be effective. We just expect the high dose to be even more effective. And it's a year-long study, very, very well powered. And so one of the things that's important for us is to really get a black and white as much as possible picture. If the drug fails, if the study fails, we really want to know that it was because of the drug, not because of a design. And vice versa, if the drug really is effective, we want to be able to capture it in a well-designed and well-executed study.
Mm-hmm. And if I remember correctly, I'm just trying to picture the curves that you had from the previous study. Like you disclosed the follow-on data, and it seemed like the patients generally got better over time.
What we've seen in the initial study was separation between linabecestat and placebo, so that's always encouraging to see. Mm-hmm. Again, sometimes statistically significant, sometimes directionally correct. What we've seen in the open label extension, where everyone now goes on drug, is patients continue to improve, and then at some stage they just hit a floor. You can't get better than mm-hmm. as best as you can, and they're maintaining that floor, and that makes us very, very happy.
Yeah. Oh, I think the curves were it might have been the responder rate. Mm-hmm. So eventually, you get like just more and more people were responding. Just like some people might not have responded at like 12 weeks, but maybe they responded at 24 or later. Was that the case?
It was the case, and again, to be completely transparent, some people just don't respond.
Mm-hmm.
I will always worry about a drug that works 100% of the time on 100% of people. That worries me a lot. But what we've seen here with lenabicim, at least in this small study, is the majority of them respond. They tend to get better over time, and then they stay stable, which is important. In all of our diseases, Max, we're looking at lifelong chronic therapy. These are diseases that will always be there. The idea is get to a point where you've either alleviated the symptoms or modified the disease to a degree where the quality of life, and hopefully because of that also the length of your life, your lifespan, has been dramatically increased.
Mm-hmm. And how big is this market, by the way?
In terms of members, so in the United States for systemic sclerosis, you're looking at about 90,000 people. If you add the EU and Japan, you get to about 200,000 people. And again, while there's standard of care, which is typically cytotoxic drugs, there's no approved drug for it. Dermatomyositis is a little bit smaller. It's about 50,000 patients in the United States. And then cystic fibrosis is the one that's best understood. It's about 75,000 patients pretty much in the world.
So, I mean, these are all going to be, you'll have orphan drug and all that stuff, because if I remember correctly, this has a bit of a history, lenabicim. If I pronounce it wrong, that's always my problem.
You pronounced it beautifully. We have orphan designation for all three of them. We have fast track, if I remember correctly, for systemic sclerosis and cystic fibrosis. And yeah, lenabicim has a really interesting history. The drug was invented at UMass Medical School. We actually own the IP for it. So it's interesting. It's not a usual licensing deal. And initially, a long time ago, lenabicim was developed as an analgesic because the thought was, hey, it's going to be yet another one of those rationally designed cannabinoids that will bind to CB1 and therefore be an analgesic. This is the most common usage of synthetic cannabinoids. What was interesting is it didn't work as an analgesic. And when we took it over, it became apparent why. This was a drug that bound very, very, very poorly to CB1, bound very strongly to CB2. So the one thing you wouldn't expect of it is classic analgesia. The flip side of it, the one thing you would expect from it, is to act on inflammation, something which typically, again, CB1 agonists, for example, have little to no effect.
Going back to the sclerosis data,
mm-hmm.
so you mentioned that like in some I guess endpoints it was positive, some endpoints it was negative. So where does it look like the drug is stronger?
So it's interesting. We actually, off the top of my head, don't think we had any negative endpoints, but we had the usual spectrum. So for example, in some of them we have very strong p-values. In some of them we don't have p-values, especially some of the secondaries that were much more exploratory. And then of course we had
just which ones were those?
Which ones were which ones?
The ones that were not. Just so we know like where the drug is.
Sure. So for example, if I remember correctly, it had a pronounced effect on the skin, but it had less of an effect on FVC. Now, to be fair, in 16 weeks, measuring changes in FVC is pretty challenging. We did see a trend towards stabilization, but it was very, very early days. In some of the PROs, for example, initially, not much of an effect on pain. These patients do experience pain. Much more effect, for example, on PROs describing disability, which have more to do with mobility of your fingers, of your extremities, etc.
Okay, great. And then now, if it doesn't have much of an effect on FVC, I know maybe it's not long enough. Cystic fibrosis, isn't that like kind of one of the main endpoints for that?
Totally different. So in systemic sclerosis, what you have is fibrosis of the lung, and you'll capture that with gradually declining FVC. And you're right, that's not the main purpose of this drug. Lung fibrosis is really, really tough. In
I know.
cystic fibrosis, it's a very different type of lung disease. It's much more wet. There's a ton of mucus, bacteria, of course. There's a ton of obstruction. And the way you capture that immediately is in FEV1. So there's basically a lot of wheezing. And the way we deal with FEV1 is CFTR modulators and DNAs. So we give them either expectorants or mucolytics. These are very sophisticated drugs that try and clear the mucus and just open up those airways. The problem, though, in cystic fibrosis is the gradual damage by the underlying inflammation. And that, interesting enough, is captured primarily through pulmonary exacerbations. Just like you get in asthma, just like you get in COPD, except they're really spectacular in the bad way. These one will land you in the hospital for 2, 4, 6 weeks. And as a side effect of that, you're going to start losing lung function as well. About half the lung function you'll lose because of lifelong, because of exacerbations. But the immediate effect of inflammation in CF max is you're in hospital, you have an acute flare of your lung disease and other aspects of your disease. It's very, very scary. It's very damaging. It's obviously, sadly, life-threatening, and it's very, very costly. One exacerbation can reach $120,000 per event in the U.S. And for that, we really don't have any drugs at all.
120,000 per exacerbation. That's like out of this world.
Yeah, but think about it. You're spending 2 to 4 to sometimes 6 weeks. You could be in ICU or in an isolation ward. Those expenses are very, very high.
Yeah, and that's probably why I think I saw there was maybe a review that came out yesterday about how there's a lot of people who are just kind of the super utilizers of the healthcare system, and people haven't figured out how to get them to utilize less. Well, if they're a cystic fibrosis patient, there's nothing you can do in terms of
a lot of choice.
Yeah, you can't change a copay and then suddenly they're going to use things less. Like that's not going to be that's a deciding factor for them. Going back to the sclerosis stage, just because you have some the large trial reading out next year. I saw that some people were a little concerned that they felt like in the placebo arm it had sicker patients.
So I remember that critique. We looked at it very, very carefully. Again, this is a small, short study, and there's only so much randomization you can do with 44 patients. Having said that, we actually felt very, very comfortable that our baseline characteristics were as evenly distributed as possible, remarkably so. And maybe it's worthwhile describing what these patients look like. The first thing is they look a lot like other patients in other studies. There was nothing that we could see that was out of the ordinary in terms of what they would look like. They're primarily women. They're all adults. This is a disease of adults. They're all typically moderate to severe and refractory. They're all on standard of care, most of them are on standard of care, which is cytotoxic drugs basically taken from the world of oncology. And then we either put placebo on top of it or the drug on top of it. And we measured to see what was happening. One of the interesting things about the primary endpoint, which was CRIS, which is now the endpoint also for the phase 3, CRIS is a very, very, very complex mathematical algorithm looking at 5 different endpoints that you put together. It's nonlinear, it's not normally distributed, but one of the advantages is it tends to be really good at eliminating a placebo effect. In fact, in the 5 studies that have used CRIS, ours was prospective, but others were retrospective or of the phase 2. All the studies except for one had no placebo response in CRIS. So it's really good at doing what it's supposed to do. And we saw, as have others seen, a very, very marked difference between linabecim and the placebo. The other thing that made us feel very encouraged was between the double-blind placebo-controlled and the open-label extension was actually a very long washout, on average 20 weeks. And what was interesting, Max, about that one was folks who had gotten who had gotten linabecim worsened during the washout. In other words, the moment you took away linabecim, they started getting sick again, or there again, the morbidity started to get worse. Then you put them back on linabecim, and the trend started to reverse again for the better. So we're very, very encouraged by that. And of course, as we're now doing a much larger study, any sort of imbalances should be smoothed out just by the sheer powering of the study. And we'll see. We're pretty excited about next summer.
Yeah, that's great. And so in terms of like competitors.
So I would separate competitors from standard of care. The standard of care are very, very, very potent cytotoxic drugs. Mycophenolate, cyclophosphamide, Cellcept, for example, etc. So that's a standard of care. It typically most of it has never been shown to work in a double-blind placebo-controlled study, and it's really toxic. The competition is interesting. So it's almost every big pharma, because scleroderma is such an attractive market, but not a single one going after cannabinoids. So they tend to take, for example, Max, they'll take a rheumatoid arthritis drug like Actemra, tocilizumab, from Roche, and they'll try it in a phase 3 study, or abatacept from BMS, Orencia, and they'll try it in a scleroderma study. And so far, a couple of them have shown actually pretty decent phase 2 data. None of them have succeeded in phase 3, partly because the endpoints were just not very appropriate. We're the first ones to use CRIS as a phase 3 endpoint, so we're very encouraged by that. So in scleroderma, we don't have a cannabinoid competition, at least not yet. For NASH, as I mentioned, for CRB4001, we absolutely have a competitor with an almost identical pathway.
And then, and you also have like a gazillion other competitors. I tweeted out a chart of that like a few weeks ago.
So those of course are sort of the phytocannabinoid companies. And the way I think of them, Max, is there certainly are, I guess there are peers to a certain extent, but remember the vast majority of them are focusing on CNS. So whether it's epilepsy, Tourette's, pain, appetite.
No, but I actually meant for NASH. So
for NASH. Oh, yeah.
Yeah, but not, they weren't cannabinoids. It was just like, everyone and their mother seems to have a NASH program right now.
Fair enough. Look, it's such an enticing indication. We will certainly not be the first to market. I mean, there's no way, unless something goes catastrophically wrong with every single other competitor. But what we find encouraging about it, and I'm guessing that's what Janssen find encouraging, is by the time, if we do make it to market, either one of us or both, it will be an additional drug or therapy focusing on a pathway which will not have been used by then. So hopefully it will have some add-on effect. The other thing that I like about not being anywhere near the first to market is the endpoints are just so tricky at the moment, and the study design, the pivotal study designs are so tricky. I'm looking forward to other people sort of breaking their teeth on that one, so that by the time we get there, it just is much more clear on what the path to success is.
Yeah, I mean, so far, I mean, I think what was it? The Intercept drug is one of the leaders, and I mean, it seems to have like pretty modest efficacy, and then it has that horrible itching.
Yeah, I mean, it's these
you can't scratch kind of itching.
Yeah, the itchy and scratchy. This is a massive indication with sort of an enormous arms race to get to the starting line or the finishing line, I guess. And so I think it's normal that the first 1 or 2 or 3 perhaps approved drugs will not necessarily be the ones that, let's say, 5 or 10 years from now will be seen as the dominant drugs. And I think, we saw this with hepatitis C, for example. Not a lot of people remember, I guess it depends on your age, that once upon a time, Vertex was a hepatitis C company, a very successful hepatitis C company. These things happen.
It wasn't that long ago.
Fair enough, because it depends on your age.
Well, yeah. For me, like, that long time ago felt like yesterday.
Fair enough.
So on just going back to CRIS, the endpoint that you're using. So is the FDA okay with that endpoint?
Yes. So we had, and it's actually a really interesting story. CRIS was our primary endpoint for the phase 2. We were the first ones to ever use it prospectively. And then in 2017, and the dates are important in this little saga, in 2017, we went to FDA in a Type C meeting and said, hey, here's our phase 3 study, and we really want to use Chris. And at the time, the guidance we got back was, look, every other study in scleroderma has always used the modified Rodnan skin score. It's a component of the Chris, but it only looks at the skin. And the guidance was very clear. Please go ahead and use the modified Rodnan skin score. You're welcome to use Chris as a secondary. Very, very recently, I think it was last month, we announced that we've gone to see the FDA and everything turned upside down. Now we have Chris as a primary, so it's back to being the same primary as phase 2, and MRSS is now back to being the secondary. And so the question is, how did that actually happen? And a bunch of things happened in a 2-year period. The first thing is a whole bunch of phase 3 studies, or phase 2 and phase 3 studies from big pharma in scleroderma failed based on MRSS, but actually had decent data on Chris. And of course, that added to the body of knowledge that we had generated that showed that Chris was much more sensitive to separating an active drug from placebo than what MRSS was. So it was a better tool. The second thing that happened was, I think just across key opinion leaders and across the field, there had been they just reached sort of a peak in terms of frustration with MRSS. These are people that devoted their entire lives to researching this disease, and they all said, look, this is just not a great endpoint.
Does it just have a lot of placebo effect in it?
It had a placebo effect that was problematic. Now, granted, a lot of these studies were not very big. Some of these studies were done in centers that perhaps they shouldn't have done it with, but MRSS is a physician-administered exam, and it's subject to all the type of biases that physician exams are subject to. And the last thing, Max, was, and I wouldn't underestimate it, the FDA had changed a little bit. You know, there's sort of a new spirit blowing through the FDA. There have been some changes in management at the FDA. And I think not only for us, but for probably a whole bunch of other companies, sort of a more openness to embracing novel endpoints, more innovative endpoints. So I think all of that worked in our favor.
Oh, great. So anything else that you'd like to add?
You know, the only other thing I would add for your listeners is I would share a personal theory that I have, which is, you know, cannabinoids, this term is so complicated and it's so wrapped up in a lot of public perceptions. But if we just look at it as purely, think of it as pharmaceuticals designed to target the endocannabinoid system, take away the plant for a second out of this equation, then I think that what people sort of perhaps underappreciate is how well developed the biology is. We now know the crystal structure. There are thousands of papers now on basic biology of the system. And that I think this coming decade will be a decade where we could see a lot, if not most, big pharma embrace the endocannabinoid system. And if that is the case, then they're going to have to either invent their own NCEs, which is always possible, or they may have to go out there and partner with folks who just have either big libraries or deep knowledge of this biology. And I think that opens a lot of interesting opportunities for across the field, and we'll see what happens. But we're really, really excited.
Yeah, you know, I'll have to agree with that statement. I think pharma, you know, will go into this area because, I mean, this is a pharma, like in the cannabinoid space, this is the pharma-friendly end of it, which is, you know, rationally designed molecules going after a target, not, you know, phytocannabinoids, which, you know, there's no IP. And even if you have IP, it's probably going to be really weak. And unfortunately, like when I was researching my cannabis sector report, which came out recently, it looked like most of the pipeline for cannabinoids are phytocannabinoids. They're just kind of, you know, mixing CBD or in THC or just doing one or the other for a variety of indications. And that's not the way to get a large pharma partner. You get one by having like a novel molecule that actually works in, you know, in a nice large trial, and they can, you know, sell for a lot of years.
Mm-hmm. That's certainly the way we look at it. We saw, I think last week, Lundbeck bought out Abide, who are working on the same pathway. They're working on it by having NCEs that prevent the degradation of endocannabinoids. I think that was Max, correct me if I'm wrong, I think it's a $400 million deal for, I think they're preclinical or they're just in the clinic.
Yeah.
So I think there's going to be more and more appetite from the buyers. And I think there is a wonderful, self-servingly wonderful dearth of targets. So, as I said, we're super, super happy.
Yeah, I mean, all you need to do is, you have Lundbeck doing it, and then you get like one more, and then they'll all start having, endocannabinoid targeting drugs in their pipelines because, they like to be like sheep.
Exciting times.
Yeah, exactly. So anyway, thank you so much for being on the show. It's been really great talking to you, and to everyone, thanks for listening, and please review us on iTunes.
Thanks, Max.
All right, great. Thank you so much, Yuval. This has been great.
Our theme music is by Hazard.